Hello, everyone. I'm Reut, a PhD student in Professor Ehud Cohen's lab from the Hebrew University of Jerusalem. I'm honored to open the next session about molecular aging. And without further ado, I'm happy to invite our first speaker, Dr. Daniel Baal from Tel Aviv University, who will talk about lamins, metabolism, and aging. Dr. Baal, the stage is yours.
לא, הוא צריך, הוא עובד איתו.
לא צריך, רוצה.
Hello, everyone. Usually at this point, I thank the organizers, but for now it will be kind of stacking the deck. So, we don't see my presentation? Great, thank you. So after all these great talks, I don't need to persuade you at the importance of studying aging. But this is important not only for the studying of the aging process. It doesn't matter which chronic disease you're interested in. Aging is the major risk factor for it. Corona made us all experts in exponential trends. So this is a logarithmic scale. So a straight line is an exponential curve. And you can see for heart disease, for cancer, stroke, diabetes, kidney disease, Alzheimer's, we're all exponentially racing towards getting it. So we need to flatten the curve. Well, what do we know about aging in humans? We know that if we manage to flatten the curve, we'll be able to delay the onset of many of those diseases, but how? So on the one end of the spectrum, we have premature aging diseases. We have a few of those, and I'll go into detail a bit later. But these symptoms, they manifest in some of the tissues, not all of them. Some of them manifest in many tissues. And interestingly enough, 3 of them, Hutchinson-Gilford progeria syndrome, atypical Werner syndrome, and restrictive dermopathy, all come from a single gene, the Lamin A gene. If we want to expand lifespan and not shorten it, in nematodes, you can put a single point mutation in the DAF2 gene, and you increase the lifespan by about 2.5-fold, depending on the mutation experimental conditions. If you add a deletion of the RSKS, the S6 kinase in C. elegans, the downstream target of the mTOR pathway, you double lifespan again. So we can get a very significant lifespan increase in nematodes. Is this relevant to humans? Well, yes. This all depends on the DAF16 transcription factor. In humans, it's the FOXO3. If we look at centenarians, the strongest signature we see in the genome is from the FOXO3 transcription factor. We want to do these experiments in humans. If you can find a PhD student who's willing to put the time, it's about 100 years, let me know. Okay, so we have the insulin light signaling pathway. What about the mTOR? Well, if we inhibit mTOR in mice with rapamycin, we extend lifespan significantly, even if we start midlife. But rapamycin has a lot of side effects. If you don't want to go that way, you can do dietary restriction. You restrict the amount of food you have. This actually depends on exactly what you eat and when you eat it. But it's a conserved pathway from yeast all the way to humans. It can be controlled. It can be mimicked and abolished by various genetic and chemical means. And like I said, it does not result from the sole reduction in food consumption, but also timing. He's just monkeying around. So it doesn't only seem longer, but there is kind of a debate in the literature whether primates on their natural diet do get benefits from dietary restriction. Don't worry about this. None of us have a diet that even remotely resembles our natural diet. Okay, so you can do dietary restriction in nematodes. And when you do, you get animals that are smaller, they live longer, and they have a smaller brood size. So our first surprising finding was that if you knock down lamin in each two, these are dietary restricted animals, you get nematodes that are significantly larger. And this effect is specific to dietary restricted animals. The effect in wild type animals, sometimes you see it, sometimes you don't, but it's really minimal. You can see the small worm here, and it becomes significantly bigger. And these small worms, they're smaller because their cells are smaller, not because they have a different number of cells. And indeed, you look at the size of the cells, and you can see the cells, they grow bigger, and you measure the pumping rate, they're still dietary restricted. So you can take dietary restricted worms and somehow get them to be bigger. Well, what is this lamin that I'm talking about? Lamin is the key component of the nuclear lamina. It's a filamentous-like structure sitting adjacent to the inner nuclear membrane. It interacts with chromatin, with many proteins here, with the inner nuclear membrane, and with the nuclear pore complex. Mutations to the Lamin A gene cause various different diseases, more than any other human gene, including, like we said, 3 segmental progeroid syndromes. So we have this gene that is key to regulating aging in premature aging, and here we see a first clue that it is somehow interacting with a lifespan regulating pathway. Okay, so we have size. What else do we have? These worms, when we feed them less, they gain less fat. Not very surprising. You can see it here in the oil red O staining. Less red, less fat. But when we knock down lamin, even though they're starved, they still gain more fat. This teaches us that actually the decrease in size and fat isn't solely due to less resources. This is a controlled process. They don't get as fat as wild-type worms, but they get significantly fatter than their counterparts. How does this work? So we wanted to see if SREBP, which is responsible for lipid biosynthesis, is involved. Specifically, we wanted to see if transcription of SREBP is involved. We looked at GFP expression from under the SREBP promoter, and indeed we saw an increase. Not very dramatic. This isn't the only explanation, but this is part of the mechanism. So we have changes in transcription. We wanted to really pinpoint where does Lamin A interact with dietary restriction. Luckily, in nematodes, lifespan regulating pathways have been mapped better than in any other organism. Several genome-wide screens have been performed, and we know which pathways regulate lifespan. We don't know all the exact details, but we know the major pathways and the major players. And if I had to take a guess, and I did have to take a guess, I would say probably the mTOR pathway and maybe the insulin-like signaling pathway. So to do that, that's a simple experiment. You take the dietary restricted animals, you cross them with animals that lack S6 kinase, and you can see the size increase from lamin knockdown is completely abolished. No effect at all. Well, the mTOR pathway works through true complexes. mTOR C1 is kind of the natural target. mTOR C1 has a unique protein important for its function, Raptor in C. elegans DAF15. So if we knock down DAF15 and lamin 1, again, no size increase. So if we have the mTOR pathway, we know that the mTOR complex and S6 kinase work downstream of lamin. Let's try to work our way up and find how this works. So starting going up, we have Reb. Knocking down Reb, we found something strange. You take wild-type animals, you knock down Reb, the animals become smaller. You take down dietary restricted animals, you knock down Reb, the animals become bigger. Why does the same protein have two different effects? Actually, the answer to that is from a paper we published a few years ago. Reb is found in two forms, GTP known to activate mTOR, and GDP, which we claim represses mTOR. In the well-fed animals, Reb is predominantly in the GTP form. You reduce it, you reduce activation. In the dietary restricted animals, Reb is in the GDP form. You reduce it, you reduce inhibition. In any case, this makes no difference for us because Reb has no effect whatsoever on the downregulation with Lamin. So Lamin acts downstream of Reb and upstream of mTOR. We also looked at other components of the mTOR like RAGA, and again, complete abolition of the effect. Other pathways like DAF2, DAF16, no effect at all. If we go even further up, AKT, no effect. AMP kinase is very critical, plays a role upstream. It does not prevent size increase. Actually, it may cause wild-type animals to be more sensitive. So it actually may increase that effect. But Lamin works somewhere in between these two. So let's zoom in on the mTOR complex 1. There's various models in the literature. This is the one we liked most from the Sabatini lab. There's a lot of support for it. So they actually show that while rapamycin, a life-increasing drug, inhibits mTOR, metformin, what it does, it acts on the mitochondria. We heard how important that is. But somehow, and we don't really know completely how, when we give metformin, the mitochondria prevents the passive entry of RAG-C into the nucleus. This passive entry is needed for it to switch from GTP to GDP form. Then it can exit and activate mTOR. Okay, so this is a model. How can we test it? We can generate transgenic animals. We knocked in a flag adjacent to the endogenous RAG-C gene. I don't know if you can see it here, but you can see in the dietary restricted animals, RAG-C is excluded from the nucleus, and you can see the quantification here in wild type, in dietary restricted versus control. And if we knock down Lamin, RAG-C can get back into the nucleus and activate the mTOR pathway. So our model is, in the wild type condition, RAG-C can enter and exit, activate mTOR. In dietary restriction, and when we're treating patients with metformin, which is now the first drug to be tested for human longevity, and in other conditions, we have the nuclear envelope preventing RAG-C from entering the nucleus, getting modified and activating mTOR, so mTOR becomes inactive. Now, we don't have proof, but we think that this is actually partially due to mechanical strain that Lamin acts on the nuclear pore complex, and it may involve phosphorylations of Lamin. We're not sure about that, but we have evidence to suggest. But the more important thing here is that we took and saw how all of these things converge together. How Lamins, known to regulate lifespan in the deleterious cases of progeria syndromes, actually interact with the conserved known pathways that regulate normal and extended lifespan in humans. What can we do next? Well, we found something because we knew from the literature where to look, but we can do this hypothesis-free, and we have proximity labeling working, and we can do this now in human tissues as well. We can label organelles with biotin and pull out and see which proteins are there. So we can see how the composition of the nuclear envelope and the nucleus change in dietary restriction, in old age. And to make it even cooler, this isn't published yet, but we actually have proximity labeling now working with two independent labels. One is azide and the other is biotin. So you can take the same sample, label one organelle with one dye, another organelle with another dye, pull them apart, multiplex them, and run them together on the mass spec. So with that, I'd like to thank, first of all, HUJI, who did most of the work on this project, the Tsur lab that helped with generate the transgenic strains. All of this work started way back when at my PhD at the lab of Professor Yossi Greenbaum, which helped a lot. I'd like to thank reviewer number 2, who's looking at us right now in Zoom. And I'd like to thank you all and be happy to take questions.
We have time for questions?
Thanks, Daniel, for a wonderful talk. Do we have time for questions? One short question.
What do you think about David Sinclair's NAD or some analogs of NAD plus in terms of, and he says that that's why he looks so young.
So the question was about analogs boosting energy level. I think his data is probably right. There is an effect. It's not a very big effect. It probably works through the mitochondria. I don't think he or anyone else knows the full mechanistic details. It's not as dramatic as really activating the pathway, because when you do all these interventions, you're always worried about feedback. You change something and then there's a feedback, and Gary Rufkund back when said, well, any toxin that you'll take, if you take it in a small enough dose, there'll be a positive effect. So we want to be kind of cautiously optimistic about these things. I think once we fully understand the pathway and see where we can turn the natural knobs and activate it the way exercise and dietary restriction and DAF2 mutants do it, we'll get a much, much better result.
Thank you.